atención y, por supuesto, si quieren hacer alguna pregunta, cuando quieran. Gracias. Gracias. Si hay tiempo, Octavio.
Sí, muchas gracias, agradecemos la excelente plática y la forma tan didáctica que dio el doctor Bolaños para su presentación.
Gracias.
Tenemos tiempo para un par de preguntas solamente.
Gracias. Tu charla sobre el metabolismo, que a algunos nos parece algo oscuro todavía. Pero tengo dos preguntas. Esta, si me acuerdo bien el nombre, es fosfofructasa 1,6-bisfosfato, que es el paso limitante para que se utilice la glicólisis en las neuronas. Sí, estoy bien, ¿no? Porque tienen esta cosa ubiquitinada.
Sí, correcto.
¿No? Okay.
FKFB3.
Esta molécula puede ser transportada de astrocitos a neuronas en alguna circunstancia fisiológica importante.
No, el metabolito, ¿te refieres al activador alostérico?
Exacto.
Podría ser, pero lo veo muy difícil porque está muy cargado. Date cuenta de que tiene dos grupos fosfatos.
Okay.
Los dos grupos fosfatos le confieren una carga negativa, una densidad de carga negativa muy grande, y yo no conozco que se sepa que hay algún transportador para esto.
Tal vez a través de conexinas.
Si hay conexinas entre las neuronas y los astrocitos.
Sí hay.
Pues,
podría ser.
No. Podría ser, ¿no?
Okay.
Es una cosa muy interesante. Pero desde luego, si es así, sería pues difícil luego, digamos, entender por qué in vivo, como ha visto Bruno Weber, las neuronas tienen menos glucólisis que las... Porque si les está cediendo ese activador alostérico, las neuronas tendrían tanta glucólisis como los astrocitos.
Sí, sería quizá en algún tipo de situación fisiológica relevante, ¿no? Sí. Podría ser.
Sí, activar la conexina para activar la glucólisis.
Exactamente. La otra pregunta tiene que ver con esto último que estabas platicando. O sea, lo que nos estás diciendo de alguna manera es que el metabolismo, o sea, la generación de especies reactivas de oxígeno en el astrocito es mucho más elevada que en las neuronas. Por lo tanto, eso les tiene como preparadas el metabolismo para ser más resistentes que las neuronas. Eso podría ser una interpretación.
Eso es lo que es muy interesante. De hecho, es hasta, a veces podría ser hasta paradójico, ¿no? Porque las defensas antioxidantes en los astrocitos son mayores que en las neuronas. Sí. Todos los niveles de las enzimas antioxidantes están mucho más elevadas. Entonces, a nosotros nos chocó bastante esto, o sea, nos sorprendió bastante estos resultados, porque lo que esperábamos era lo contrario. Y de hecho, lo que parece que está en la mente de casi todas las personas es que las neuronas producen más ROS y por eso son más susceptibles a los ROS. Sin embargo, nosotros lo que vemos no es eso, es lo contrario. Lo que pensamos es que podría haber una velocidad de recambio de ROS mayor en los astrocitos que en las neuronas. Es decir, que la formación de ROS es mayor, pero la velocidad de descomposición también. Nosotros lo que estudiamos aquí es la es formación de superóxido. Claro, es difícil saber cuál es la concentración de especies reactivas de oxígeno en el estado estacionario. Eso no es fácil de determinar. Por lo menos a mí no me resulta nada fácil, ¿no? Lo que nosotros proponemos es que hay más formación de superóxido y más degradación de superóxido. Se tiene un turnover alto, mientras que en las neuronas eso no sucede. Y esto es compatible con un papel señalizador. Como casi todo lo que se sintetiza mucho y se degrada poco, como todos los segundos mensajeros, es que tienen alguna función específica alterando alostéricamente pues alguna función. Y eso es lo que nosotros pensamos y estamos estudiando.
Y los ROS en general
se sabe que son señalizadores, exactamente. Pero bueno, nosotros lo que estamos viendo es que si en los astrocitos la función que tienen es esta, ¿no? Están listos como, efectivamente, están preparados como para que los ROS en los astrocitos sea una, como tú dices, una... Efectivamente, algo cotidiano. A ustedes.
Una pregunta más.
Ok. Me llama mucho la atención esto de que las neuronas tengan... Me pregunto si sabes algo acerca de la cuestión evolutiva. No sé, por ejemplo, si el es como un gasto de energía que no sé unos dos y también me pregunto acerca del desarrollo de los mecanismos, ¿no? Y durante el envejecimiento estas cosas cambian, entonces hay más entre las neuronas.
Las dos preguntas. Sí.
También por ejemplo, la corteza
Mónica, muchas gracias. Las tres preguntas son enormemente pertinentes y relevantes. Lo único que no te puedo dar respuesta porque estamos en ello. Es decir, son recientes estos resultados y no tengo un grupo tan grande como para dar respuesta a todo lo que me estás diciendo. Pero sí que puedo hipotetizar, ¿no? Sí que sé que hay algunos grupos que están interesados en ver la expresión de la PFKFB3 en diferentes áreas del cerebro. Lo han visto también en astrocitos y cómo eso afecta a la protección de las neuronas. La primera pregunta que me has hecho es que ¿por qué tienen la maquinaria? Bueno, pues la glucólisis es una maquinaria interesante, ¿no? Porque no solamente sirve para formar piruvato y luego energía, ¿no? En las neuronas, es decir, en cualquier célula, los intermediarios de la glucólisis están interconectados con muchas vías metabólicas. Síntesis de fosfolípidos, por ejemplo. Necesitas dihidroxiacetona-fosfato, ¿de acuerdo? Entonces, eso es uno de los ejemplos, pero hay muchos ejemplos más. Entonces, modular, digamos, tener la maquinaria preparada para utilizarla no como una fuente energética, sino para sintetizar, para conectarlo con otras vías metabólicas, puede ser interesante. ¿Cómo está regulado? No lo sé. Lo que dice Pavel me parece una idea fantástica. Si a través de las conexinas pasara la fructosa 2-bifosfato, eso sería un modo de regular endógenamente esto, ¿no? Pero habría que estudiar. Hiciste dos preguntas más, una... Sí, no lo sé, no nos ha dado tiempo, digamos, a verlo. Pero sí, es decir, el envejecimiento, no sé, ¿tú qué esperarías ver? ¿Un envejecimiento? Podría ser. Y el de la neurona menos capacitada para fabricar energía. También, exactamente. Sí, yo no lo sé porque no lo sé si eso está hecho, ¿no? ¿De acuerdo? Pero yo sé que hay controversia, hay un tema interesante de los astrocitos en humanos, y es que estuve hablando con José María Verdugo, está en Valencia, el que vio la primera célula madre en humanos, lo publicó en un Science.
Trabajaba
Sí.
Con este chico que está en San Francisco, ¿no?
Sí, exacto. Bueno, pues una vez yo estaba hablando con él y me dijo, dice, no, esto que me acabas de decir, claro, los astrocitos pueden perder su función en el envejecimiento, porque y se dice en humanos, además, los astrocitos tienen una morfología bastante más fibrosa que lo de los ratones. Pero claro, la morfología de los astrocitos de los humanos está estudiada en adulto y es más, en viejo, que son la mayoría de los que ceden sus cerebros, ¿no? Han muerto. Pero no en niños, ni en gente muy joven. Eso no es lo habitual. Sin embargo, nosotros estamos acostumbrados a hacer toda la inmunohistoquímica en ratones jóvenes. ¿De acuerdo? Yo le dije, la verdad es que se sorprendió. Y dijo, pues la verdad es que no lo sé. Voy a mirar hoy cómo están los astrocitos en niños. Y algunas muestras por ahí tiene, no sé si lo ha hecho ya. Es decir, que no lo sé. El tema de los astrocitos es un tema muy interesante y cómo controla el metabolismo de las neuronas. No te voy a quitar más tiempo, porque sé que hay muchos speakers.
Okay.
Bueno,
muchas gracias. Nuevamente. Pues, si tienen alguna otra pregunta, pues yo creo que con un correo, ahorita en un breve receso, se la pueden dar al doctor Juan Pedro. Aquí está. No, tenemos en la diapositiva.
¿Cuál es?
Tú, a ver. Esta la minimizamos. Y Adrián es la de... ¿Cuál es la tuya? ¿Esta?
¿Pero cuál es?
Esta. Gracias. Bueno, damos paso a nuestro siguiente invitado. Él es el doctor Adrián Rodríguez Contreras. Él es biólogo de formación, realizó sus estudios aquí en la Universidad Nacional Autónoma de México. Posteriormente hizo su doctorado en la Universidad de Cincinnati y tiene varias estancias postdoctorales, una de ellas en la Universidad de California, en Davis, y otra en Holanda. Actualmente el doctor Adrián Contreras trabaja en el Departamento de Biología en la Universidad de Nueva York, y en esta ocasión pues nos va a presentar un trabajo titulado Development of the Neuroglía Vascular Unit. Entonces, pues agradecemos mucho la presencia del doctor Rodríguez Contreras. Muchas gracias por estar aquí. Gracias.
Buenos días a todos. Antes de empezar, quisiera agradecer muchísimo la invitación de Octavio y de Daniel a participar en este primer simposio. También espero que este sea un evento que pueda ser repetido en años próximos. Yo, como Juan Pablo, ya llevo como 20 años de no venir a México. Bueno, no tanto, sí he venido casualmente, pero es la primera vez que doy una plática en Ciudad Universitaria. Y como Octavio menciona, yo soy graduado de la Facultad de Ciencias, entonces es una invitación muy emotiva. Y a petición de Daniel, me voy a cambiar a inglés, porque también creo que es importante que para todos ustedes que están haciendo cursos de posgrado, ¿no?, que empiecen a practicar su inglés, porque la ciencia es un asunto internacional y la mayoría de las ciencias se hace en inglés, pero eso no quiere decir que el conocimiento no pueda ser también transferido a otros idiomas, ¿no? Entonces, well, what I'm going to do now is I'm going to switch. And my talk, when I talk about the neurogliovascular unit, I want to emphasize that to understand the normal physiology and pathology of the brain, we have to take into account that we have to understand multicellular interactions, okay? And the idea here is that we are not there yet developing the methods that would allow us to actually look at the function, the coordinated function of the brain in terms of cellular activity, okay? So now my laboratory is interested in studying neuroplasticity in the context of postnatal development. And very recently, we have become very interested in the neurophysiology or of neurovascular physiology in neonate animals, okay? So my first slide, Juan Pablo has already pointed out that there is a very important metabolic coupling between neurons and astrocytes. And astrocytes are also very interesting because they can couple with blood vessels, okay? And as you can imagine then, there's a so-called neurovascular coupling that basically links the activity of the neural tissue to changes in blood flow. And why is this important? Because all those pathways that Juan Pablo has told us about rely on the presence of nutrients, and the oxygen, right, is coming from the bloodstream into the brain tissue. So neurovascular coupling is one of the hypothetical functions of the astrocytes. The astrocytes might have a very important role here as intermediaries between the neurons and the blood vessels. And related to this is also the fact that the vasculature of the brain is different from the vasculature in other organs. It contains a structure called the blood-brain barrier that limits the flux of certain compound solutes that are present in the bloodstream, but it allows the glucose and the oxygen and other things that are important not only for the metabolism of the cells that live in the nervous system, but also that act as signaling molecules. Just think about the endocrine signaling that goes on physiologically. Now, what I want to indicate here is also stress, is that the interaction between the astrocyte and the blood vessel is not random. It seems to be focused on the arteries and the microcapillaries that are associated with the oxygenated blood that comes from the heart, okay? And much less so with the veins that carry out the deoxygenated blood, okay? Now, this interaction is not new. In fact, here is our friend Pío del Río Ortega, who is one of the pioneers in the study of Neuroglía. He was one of Ramón y Cajal's students. In 1920, he made this beautiful drawing where he already indicates that astrocytes form these structures called end feet that wrap around the blood vessels. And right here, we have a more modern picture that we can obtain with microscopy, light microscopy, fluorescence microscopy. The advantage of this method is that now we can use, for example, antibodies that recognize specific molecules on the blood vessel. And here, the blood vessel is dyed in a green color because there is an antibody that recognizes a molecule called laminin. And this particular blood vessel is very rich in laminin, so it is labeled. And in cyan, we have the processes of astrocytes that are expressing a protein called GFAP, glial fibrillary acidic protein. Okay. So basically, my laboratory is taking advantage of this very long tradition of using light microscopy to illuminate, to reveal the structure of these interactions. Okay. And importantly, people have gone on to perform analysis at an ultrastructural level using electron microscopy. And these studies suggest that the interaction between astrocytes and blood vessels is very intense. And so in adult animals or in adult human brains, the arterioles and microcapillaries that are associated with these vessels are fully covered. Almost 99.9% of the surface of the vessel is covered by the astrocyte membrane. Okay, and just to give you an idea, right here we have the arterioles that have a relatively large diameter, and here we have the microcapillaries which have a smaller diameter. And basically, what you see here is the endothelial cell is right here. Here is the lumen of the vessel. So this is a cross section across a vessel. And right here is the end feet of one astrocyte, is another one. And both arterioles and microcapillaries have these contacts. The contacts between the astrocyte and the blood vessel are not directly on the endothelial cell, but there is a cellular layer in between. And the difference between the arterioles and the microcapillaries is that in the arterioles, we have a single layer of smooth muscle cells. And in the microcapillaries, we have maybe not a continuous layer of cells, but called pericytes. And these two kinds of cells have been demonstrated to have contractile properties. So the idea here is that by changing the diameter of the vessel, one can control the flow of nutrients or the flow of blood, right? So a dilation of the blood vessel would result in an increase in the local blood flow and presumably change in the perfusion of the tissue. Now, this is important because one of the limitations today is basically to study the permeability of these capillaries and how this changes in terms of the activity of the neural cells. Okay, so those are sort of the main questions that we are interested in studying in my lab. And why is this important? Well, just think, for example, about biomedical imaging. And here I'm talking about techniques that use magnetic fields, right, to measure changes in the oxygenated levels of blood as a human being is performing a perceptual task or engaged in some kind of mental function. And so basically you see that parts of the brain light up. Well, it turns out that this is an indirect method of measuring brain activity. In reality, what we are measuring is the changes in blood flow that are associated with these increases in oxygenation. So understanding the cellular mechanisms that go on during neurovascular coupling are very important for this area. It's a direct application in medicine. It's also very important for developing strategies to deliver drugs to the diseased central nervous system. So it turns out that the presence of this blood-brain barrier that prevents pathogens and toxic substances from entering the brain, it's also preventing drugs that we design to treat neurologic disease, okay? So, so understanding how the permeability the mechanisms of permeability is very important for this context. And lastly, it's been shown that under pathological conditions, there is basically a disruption of the integrity of the blood-brain barrier. And basically, this can happen during stroke. It can happen in neurodegenerative conditions like Alzheimer's, and other examples are like Parkinson's, multiple sclerosis. These are all examples where at some point during the pathology of this disease, we observe changes in the permeability properties of the blood-brain barrier, okay? So today, I want to tell you two stories. My laboratory has always been interested in studying the development of neurons, but eventually we could not ignore the presence of the glial cells, and in fact, the presence of the perivascular cells that are associated with the blood vessels. So the first story is work that basically we're just going to tell you how we got interested in neurovascular physiology. And this is work that was done by Aminat Saliu, who was a master's student in the lab, right? So then for those of you who are thinking about joining the master's program, go ahead and do great things. And Aminat was helped by my then lab manager, Shanadis, and by a high school student, Kamila Kudelska. So this is basically a team of very young women, right? It's also very important because for a long time, science and engineering has been dominated by males, right? So all those women who are here, tell your friends to come and do science. And the second story is basically a follow-up of our initial observations, because one of the things that Juan Pablo also mentioned repeatedly during the talk is that some of these processes might be very dynamic, and we still cannot image how dynamic they are and what is the time scale of these changes. But one of the things that we believe is happening during development and probably during the time course of a pathology is that the astrocyte-vessel interaction is changing, okay? So this might be a dynamic process. I still cannot show you direct evidence of this, but I'll show you some of the data that we have that point towards this interpretation. And this is largely work that has been led by Ling Jiangxi, who is actually in the audience today. So if I say something that makes no sense, just go ask Ling Yang, who's the real expert, and a group of undergrad and graduate students in my lab. And then at the end, I will just tell you a little bit about sort of the new directions that the lab is taking with these studies. Importantly, just as Juan Pablo mentioned, please stop me at any point in time. If you have a question or if something I say is not clear enough or you want me to repeat it, okay? So everyone is okay? Following up? All right, great. Okay, so I will start just by telling you that my initial interest to study the neonate brain started in the brainstem, okay? And one of the models that we use is the rat auditory brainstem. And here's a cartoon that shows you the initial steps in the synaptic pathways that connect here the cochlea to these groups of cells located in the superior olive of the brainstem. So we have hair cells that are the sensory cells of the inner ear that connect to the spiral ganglion neurons through a chemical synapse. These spiral ganglion neurons then make a synaptic contact with numbers of different cell types in the cochlear nucleus. Now, it turns out that in the cochlear nucleus, there is a subpopulation of cells called the bushy cells. And there are different flavors. You have the spherical, depending on the shape and all these things. But the important thing here is that these bushy cells make a very long axon that crosses the midline. So this is another example of a sensory pathway that has a contralateral representation, for those of you who have already taken some neuroanatomy courses. And here we have a series of nuclei that integrate information coming from the two ears, okay? And the functional relevance of these interactions is thought to be in interpreting the cues that allows to locate sounds the source of sound, okay? So how can we tell that the sound is coming from the left side and from the right side, from in front? Well, these are the neurons that are presumably coding the information present in these cues, okay? Now, I'm not going to go into much detail about the neurophysiology, but just to pinpoint that there's a structure right here. It's a bean-like shaped structure called the medial nucleus of the trapezoid body. And because the name is so long, I'm just going to call it the MNTB, okay? So forget about what the long name is. But what this small group of neurons allows to do is that we can identify it very well in different animals at different ages and so forth. It also contains a group of highly specialized synapses. And basically here, one of the things that makes the auditory system interesting for neurophysiologists is that processing sound requires high precision, okay, of synaptic signaling. And it turns out that some of these giant synapses that are present in the auditory system are amongst the fastest synapses ever studied, okay? One of these synapses is the so-called calyx of health. That is this giant structure. And later in the symposium, Danielle is going to tell us more about the calyx of health and astrocyte function. And importantly, just mentioning the work of Danielle, Danielle has been a pioneer in demonstrating that there are astrocyte neuron interactions in this particular brain region, okay? But very little is known about the development of these interactions. And we were also very interested in following up, trying to expand these studies into neurovascular physiology. So this slide just shows you one of my earliest publications when I still was a postdoctoral trainee. And when I was working in Holland in the laboratory of Gerard Borst, and when I got there, he said, you know, Adrian, I have this very strange data set that if you look at the structure of the MNTV in a newborn animal, so the day of birth is P0, you can already see that there are neurons there. They form, well, there are cells that they form this bean-shaped structure. And you can actually see it growing. And you can quantify the number of cells at different ages, and then you see this increase. This is so strange because there are these studies by Bayer and colleagues that show that the neurons are born in the embryo, okay? So why is this structure, why does it continue to grow? Can you figure it out? And of course I didn't figure it out, so he fired me and then I had to find a job, right? But this is what we did when I got to New York. So I charged Aminat and her team of lab mates to figure it out. And then basically what she says, well, there must be a progenitor cell, and this progenitor cell must have the property of cell division. So this is a proliferating cell. We don't think that the neurons are going to do that. Juan Pablo already mentioned it. And this progenitor either can generate glial cells, or it can generate maybe neuron cells. So maybe, you know, those earlier studies were off track, and maybe we have some neurogenesis going on. But certainly, maybe we can have some gliogenesis. So what she said is, okay, so people have been using synthetic nucleotides like BRDU, which one can inject in living animals. And these nucleotides are somehow permeable through the blood-brain barrier and make it into the brain tissues. And those cells that are dividing, proliferating, don't care of whether this is an exogenous molecule or an endogenous molecule. Duplicate their DNA, they incorporate it into the DNA, okay? And so it turns out there's a newer version of the molecule called EDU. This is the name, it's a long one. But what this allows you to do is it contains these funny chemistry, these links that you can do click chemistry. And basically this is a copper-mediated reaction. And what one can do is place a fluorophore, like a fluorescent protein, like a fluorescent molecule, And basically, you can link the fluorescent molecule to the nucleotide. And if you can do this, then it's a more direct way to measure cell proliferation, because it's binding, it's reporting the incorporation of the nucleotide directly into the DNA. Unlike the BRDU technique, where one has to use an antibody to detect the incorporation of the nucleotide. Okay, so that's just a technical detail. And basically, what Amina decided to do is to focus first on the activity of the progenitor cells. So the experiment is very simple. You inject a solution containing EDU into an animal, and you wait 2 hours, enough time for it to diffuse through the bloodstream, reach the brain, and incorporate into the cells that are dividing. And basically, 2 hours later, you sacrifice the animal, extract the brain, process for histology, and look at what's going on. So what this micrograph is showing is a triple-labeled experiment where we are using NUN. It's an antibody that we are using as a marker for neurons. We know that this protein is expressed in neurons. So you see these blue cells right here. In cyan, in magenta, we are using an antibody that detects a protein called S100 beta. Turns out to be a calcium-binding protein that is highly expressed in the cytoplasm of certain glial cells, like astrocytes. And you see these cells in between the neurons. And then here are in green the EDU labeled cells, which you can see. And so this image, I think, is beautiful because we're showing the existence of 3 kinds of cells that they are completely non-overlapping. Okay, these markers are not shared in these cells. Okay, and basically what we noticed, this was taken from a 10-day-old rat, is that what we noticed is that as we did the experiment at different ages, okay, here E stands for embryonic day 21. The rat has a gestational period of 22 days. So this is a day before birth. Basically, we see changes in the density of these proliferating cells. And I mean that quantify this. So here's the density of EDU labeled cells at different stages during development. And what she noticed is that very close to the day of birth, there seems to be an increase in the proliferating activity of these cells that is sustained until about P10, when it drops dramatically. And the arrowhead here shows an important, how do you call, like a landmark in development. I didn't say this, but rodents, certainly the rats and mice that we are using for our experiments, are altricial animals. That means that they have a very prolonged postnatal development. And these animals are born deaf and blind. And part of the reason they are born deaf and blind is because their eyelids are fused and their ear canals are clogged with tissue. So there is a time that takes about 10 or 12 days or so for when the eyelids become unfused, basically, and the animals the eyes can detect the light, right? And the ear canals open. And then, of course, those are not the only reasons why these animals cannot see or hear, but those are important steps. And so this is the time where the opening of the ear canal is happening. And why is this important? Because number one, it suggests that whatever is happening before this time in development is independent of sensory experience. And this is one of the topics that my lab has been very interested in. You know, how does the environment affect the development of the brain? So this suggests that this might be independent of auditory experience. But we also knew that the neurons of the MNTB are relatively active during these pre-hearing stages of development. And I'm not really going to go into too much detail about how this activity happens, but maybe you are familiar with retinal waves in the visual system, where there's a similar process of so-called spontaneous activity that is not driven by a sensory stimulus, but is generated within the central nervous system. Okay, so we were very interested because this might be related to the activity of the neurons, and it might be related to the beginnings of the neurovascular coupling process that I described at the beginning. So one of the things that we actually did in the lab in a separate study is to show the time course of the opening of the ear canal. So what this slide is showing is some images of X-ray tomography. So here is the raw data, and here is some three-dimensional reconstruction. So the tomography allows you to look in 3 dimensions to the changes that are happening. So if you look at a P15 rat, here the white signal is the bone, the gray signal is the soft tissues, and the black signal, the dark signal, is air, okay? So here you can see that there is a fully formed auditory canal, and right here is the cavity of the middle ear. If you look at a P11 animal, these structures are not present. At P12, there are very interesting 2 tiny bubbles that we think are very close to the ending of a structure called the Eustachian tube that connects your mouth, the cavity of your mouth, to the inner ear. So like when you are going on an airplane and your eyes get clogged, right? What do? You kind of like crack your jaw, right? And what you're doing is you are releasing the pressure that forms in the middle ear cavity. So we think we are catching up an inside-out process of cavitation, right? So if you look at P13, the middle ear cavity is already formed, but there is no ear canal. And then by P15, you have a fully sort of a structure. And I believe this is the first time someone actually tracked the development of the process of cavitation. So this is, my point here is that imaging is great. It allows you to see things that are very difficult to do with histology, by chopping the brain into pieces, right? Okay, so what we then did is try to do some kind of experiment. Back in the day, frankly, I'm not really trained as a molecular biologist, so I cannot really make use of the fancy tools that molecular biologists use. But what we did is a very simple experiment. We took pups and we performed bilateral lesions of the inner ear. And we did this because we thought, okay, so maybe there is something that comes from the ear that is related to the activity of the proliferating cells. And this has to do with a bunch of previous studies that showed that when you perform inner ear lesions, cells in the auditory structures in the central nervous system begin to die. And this process is actually age-dependent. It happens mostly in newborns, but not in older animals. So there seems to be a sensitive period during development where when you have damage to the ear, this affects cells in the brain. So we wanted to see if this had also an effect on the putative non-neuronal cells. Okay, so far I haven't really shown you any evidence of what these progenitors are for. But basically what we did is we did bilateral lesions. We waited about 24 animals for 24 hours for animals to recover. And then we performed the EDU injection. We wait 2 hours and we process for histology. So now we are looking at what happens to the activity of these progenitor cells after in response to the lesion. And so here are some examples. These are examples that show a sham operation. It's basically we expose the ear, the middle ear cavity, but basically we don't perform a damage to the structure. Okay, we just glue back the skin and let the animal recover. Here's an example of a bilateral lesion. Here you see a little bit of blood shown in red. And here is another experiment where we did only a unilateral lesion. And this is probably important because then here we can compare, we have one side of the brain that supposedly could be affected by the lesion, but we have the other side that we can use as a control, okay?
But you have to adjust, you have lateral connections.
Right, but not to the MNTV. So let me just clarify, the connection to the MNTV is strictly contralateral. So there are some nuclei that get bilateral connections, and we haven't had a time to look into those, but we focus on the one that contains only a contralateral connection. That's a great question. Thank you for doing it. Okay, so here's basically the result. when we perform the experiment, the sham operation at P2, this is very young animals. Here is the density of cells. Here is the bilateral lesion. We don't see much difference between the density of cells. But if we perform the experiment at P13, here's the sham operated animal, and here's the bilateral lesion, we see a dramatic reduction in the density of the proliferating cells. And basically here is just a control unilateral lesion that we did perform at P10, where basically the control side, the side without the damage, here's the density of cells. In the lesion side, on the lesion side, we saw a slight increase, but it turned out not to be statistically significantly different from the other one. So basically, here's the summary of many different experiments, and here's the density of EDU cells, the age at which the lesions were done. In black, we have the sham data, right? And in white circles right here, we have the bilaterally lesioned animals. So here is the data P2, no significant change. But between P5 and P12, we see a dramatic reduction or a significant reduction in the density of proliferating cells in the MNTB. And here's the data for the unilateral control, which didn't turn out to be very different from the sham animals. Okay, so basically what we found then is that somehow the proliferation of cells in the MNTB is linked to something that is happening in the inner ear. That was the main result. So what we wanted to do next is find out what these progenitor cells are doing, okay? And so we have started a series of experiments where we now inject EDU and basically wait a longer period of time before we sacrifice the animal. Several days, from 3 days up to 5 or 6 days, right? And then we ask these cells that are labeled with EDU, what other cell markers do they express? Do they express markers of neurons? Or do they express markers of glial cells? Or do they express markers of endothelial cells and so forth, okay? So here is some of the results. Here is an experiment where the EDU was injected at embryonic day 20. So the injection was when the pups were still inside the mother, and the data was analyzed 1 day after birth. So here you see in cyan cells that are expressing the glial marker S100 beta. In green, the proliferating cells, and in white, cells that express both markers. Okay. So here is the result for this experiment. Here we inject at P1 and analyze at P4. Here we inject at P6 and analyze at P9. And basically, what we quantified here is the density of cells that were double labeled with EDU and S100 beta, because that is basically is meaningful because we know that the cells that were proliferating on the day of injection, we know that they survive all the way to the day that when we analyze the tissues, right? But they were also expressing some marker that we can assign to them, so that we can assign an identity, okay? These ones, of course, there are cells that are EDU positive, but they are S100 beta negative. Those cells, we don't know what they became. We don't even know if they are still in a proliferative stage of activity. We have cells, obviously, that are S100 beta positive, but EDU negative. Right. And so for this particular study, we just focus on astrocytes. Exactly. So there must be NG2 cells, microglia, and it turns out that we have looked also at endothelial cells, but we haven't looked at these other kinds of cells that you are pointing out. So, but that is a great point. So basically, what we found in terms of putative astrocyte development is that the majority of these cells express the astrocyte marker, right? Or a big density of these cells express the marker at perinatal ages. And as the animals grow older, somehow these cells are no longer turning into astrocytes. So what you point out is very logical. It's very likely that there are other cells that are being formed. In fact, this correlates with the increase in expression of a protein called MVP, called myelin basic protein. And we think that maybe other kind of cells that are being generated are the oligodendrocytes that will form the myelin that is also very important for axon, the transmission of action potentials along axons, which is also very important for this particular neuronal circuit. Okay, so basically what I showed you is that central cell proliferation is reduced by peripheral lesions. We don't really know the mechanism, and here what I'm talking about is a very different level of interrogation of the system, right? Because we don't know if we are disrupting normal development. For example, by damaging the inner ear, we don't know if we are taking away a signal that is necessary to maintain proliferation, or we don't know if we are actually generating a pathological condition, right? Something like inflammation, where now we are inhibiting directly the proliferative activity of the cells. We know that some of these cells generate astrocytes, but we also don't know if these astrocytes are actually affected by the lesions, right? That is something that we are interested in following up. What we did next after this initial experiment, and this is largely data that has not been published, is we asked what happens to the activity levels of the neurons in the MNTV? Because we had done previous studies where we actually showed that the cells were active. And the experiments that we performed